Hi there, my name is Naomi Joseph, and I'm going to be giving a review of the paper Virtual Core Flow Cytometry by Jared Swalwell and colleagues from the University of Washington. I'm going to start off with a brief background in the history of flow cytometry, talk about the problem statement they were trying to solve, what the approach was that they took, the results that they collected, and conclusion from this paper. Flow cytometry has been around for over 20 years, helping to make advancements in many fields, including oceanography, stem cell biology, immunology, and many others. In oceanography, flow cytometry is useful for distinguishing changes in plankton throughout the seasons and the ocean's regions as a useful assessment for the ocean's climate change. In immunology, flow cytometry helps with immunophenotyping or quantifying the different types of cells in a heterogeneous sample. Although there are many applications of flow cytometry, the foundation of its mechanism is similar. A sample of cells or particles is injected into the flow cytometer. From here, the sample is fed through a sheath fluid in such a way that the cells or particles of the sample are in a single file line passing through the opening of this center tube. This way, when the sample crosses the laser, the light scattered and detected is the result of a single cell or particle. The light is scattered in two directions, both forward and sideways. The forward scattered light is detected by the forward scatter detector and potentially the fluorescent detectors as well, depending on the sample type. The side detectors also recognize the scattered light and record the voltage from the cell particle. The output data generally looks like populations of cell types or particles types with the axes of the graph providing information about characteristics of the distinguished cell or particles. One of the important criteria for flow cytometry is properly positioning the particles or cells in the sample stream in front of the laser. The most common way to do this is with a hydrodynamic focusing via a sheath fluid. However, this material is not so readily available outside of a well-equipped laboratory, and this paper aims to describe a way to circumvent this problem. The authors of this paper designed a virtual core flow cytometer, or VCFC, which detects the position of particles that pass near the center of the laser. It does not require a special sheath fluid and could be applicable in various settings and fields of research. The fundamental mechanism of this type of flow cytometry borrows from the imaging properties of confocal microscopy. The underlying concept is that when light shines in a certain orientation on an object, it will pass through a pinhole in the imaging plane. However, if light shined is disoriented, it will be scattered elsewhere. In the diagram above, light entering from the left is a stream that is focused via a 50-time microscope objective onto a pinhole mirror aperture. This aperture in the middle is affixed with two prisms on either side of its opening and is also placed in the middle of two orthogonal detectors. If the virtual core produces scattered or fluorescent light that is focused precisely on the pinhole, it will pass through to the FSC camera. However, light not positioned properly and coming from outside the virtual core will generate a blurry image on the mirror and thus send light to the orthogonal detectors. The particles that are positioned properly in front of the laser are able to be detected and recorded only from the FSC detector and not from the orthogonal detectors. This figure shows how one is able to distinguish particles that are in the virtual core and those that remain outside the virtual core. The first thing to notice is these are bivariate plots comparing data from the orthogonal detectors D1 and D2, normalized with corresponding data from the FSC detector to correct for large particles outside the core masquerading as small particles inside the core. 1 and 6 micron fluorescent beads were fed as samples into the VCFC, and the stream was swung side to side to test how well the detector could distinguish particles directly in line with the laser. Particles within the virtual core lie along the diagonal of this graph, as shown in the ellipsoid on the left graph. The ellipse on the right also indicates the same population of particles that lie within the virtual core. The curves on the graph on the left represent the side-to-side motion from the initial sample stream of particles. Another test conducted in this paper was fluorescently labeling CD4 and CD8 of marine T cells and using the fluorescent detectors after the forward scatter detector of the virtual core flow cytometer. They compared results with the traditional flow cytometer as well as the results of these measurements, and those are shown above. The three plots represent bivariate data where the x-axis represents CD4 expression and the y-axis represents CD8 expression. The first graph on the left displays the results from a traditional flow cytometer with sheath fluid core. Three populations of cells are easily distinguishable: CD4, CD8, and cells with both labels. The second and third graph represent the same data but using the VCFC approach. The middle graph is the results with the PSD gate inactive, whereas the right graph has the PSD gate active. The PSD gate discriminates between cells within the stream and the 1.4% of those same cells that are actually correctly positioned for measurement. The most right graph is able to distinguish the labeled cells because it only considers cells positioned properly for analysis. The authors discuss that this graph clearly shows their approach is able to recreate both general characteristics of the graph from the traditional flow cytometry approach as well as quantitative results. The same study was mentioned above was replicated, but using raw seawater as a sample this time. Phytoplankton in the seawater can be classified by their expression of chlorophyll and phycoerythrin. The graph on the left represents the data collected from a traditional flow cytometer, and the graph on the right represents the data collected from the virtual core flow cytometer. Again, it was concluded that general characteristics and quantitative measurements of particle concentrations were similar between the two methods of analyses. In conclusion, the authors of this paper believe that the virtual core flow cytometer was able to greatly simplify a once complex system requiring large amounts of sterile sheath fluid into a technology that is based on a PSD creating a virtual core and less of a requirement of using sheath fluid. This makes their new approach more applicable to field laboratories, such as in oceanography or more environmentally geared research. A few next steps include experimenting with various fluorescent probes and using different shapes in the pinhole shown above, such as an H shape, to increase the contribution from orthogonal position sensitive detectors. This might help with discrimination of particles process following data collection. And these are my references. Thank you very much for your time and attention.